Greetings and welcome to the United States Transhumanist Party Virtual Enlightenment Salon. My name is Gennady Stolyarov II, and I am the chairman of the U.S. Transhumanist Party. Here we hold conversations with some of the world's leading thinkers in longevity, science, technology, philosophy, and politics. Like the philosophers of the Age of Enlightenment, we aim to connect every field of human endeavor and arrive at new insights to achieve longer lives, greater rationality, and the progress of our civilization. Greetings, ladies and gentlemen, and welcome to our U.S. Transhumanist Party Virtual Enlightenment Salon. Today is Sunday, November 2nd, 2025, and we have a fascinating conversation in store for you about an experiment that will help us to understand and bring about rejuvenation in rats and provide some insights to hopefully in the not too distant future also contribute to rejuvenation in humans. So joining us today is our distinguished panel of U.S. Transhumanist Party officers and members. I am Gennady Stolyarov II, chairman of the U.S. Transhumanist Party. With me is Art Ramon Garcia, our director of visual art. Also joining us is David Wood, who is the founder of London Futurists and the executive director of the LEV Foundation, which is the Longevity Escape Velocity Foundation, also doing excellent rejuvenation work in mice via the robust mouse rejuvenation studies. And our special guests for today are Nicolás Cherñavsky and Nina Torres Zanvettor, who are both the co-founders of the Rejuvenation Science Institute in Brazil. The Rejuvenation Science Institute has the mission of carrying out and promoting scientific research in the field of rejuvenation, longevity, and aging. Its ultimate goal is to develop rejuvenation therapies to eliminate the limit that aging imposes on human lifespan and prevent the age-related diseases such as cardiovascular diseases, cancer, and Alzheimer's disease, which kill 110,000 people worldwide every day at the very least, and this death toll is just horrific. It's ongoing and it needs to stop. So I invite Nicolás and Nina to discuss their efforts in seeking to replicate a famous rat rejuvenation experiment that was undertaken by Professor Harold Katcher, in which the researchers extracted small extracellular vesicles from the blood of young pigs and injected them into old Sprague-Dawley rats, reporting that they managed to rejuvenate the animals according to a series of parameters, including memory, grip strength, blood markers, senescent cells, and others. And actually, what was remarkable about Harold Katcher's experiment is it led to a world record in rat longevity. Sima the rat, who died in early 2023, reached the age of 1,464 days, which is just a little over 4 years, about 4.008 years. And no rat has lived, at least in any sort of recorded publication, for over 4 years prior to that time. So I'm really fascinated by the attempt to replicate this experiment because that is what science is all about. It's about replicability. So Nicolás and Nina, please tell us more.
Well, hi everybody, and thank you for inviting us. So yes, we heard about Harold's research in early 2020 because he published an article in bioRxiv. And in the article, they reported a 54% epigenetic rejuvenation as measured by Steve Horvath, Dr. Steve Horvath, who is the creator, the inventor of the epigenetic clock. So it was very impressive. And so far, there is no reported epigenetic rejuvenation as extensive as this one. And basically, I don't.
Well.
You want to tell the whole story, or?
Well, a bit. First, hello everybody, the audience, the people present here. So we had a background on the rejuvenation field before 2020. we had and we have a publisher that edited and translated and published books of the rejuvenation field. And so in that moment, 2020, we were indeed a bit tired of the lack of success of the rejuvenation therapies that existed at that time. And in fact, to be honest, even the theoretical part of the aging theories weren't really good enough, in our opinion. They didn't make total sense. So when that article was published, it was very important because despite it was on bioarchives, Steve Horvath was the first author. And I thought Steve Horvath wouldn't put his name as first author in something clearly wrong, you know. But of course, that doesn't mean it was right. But that was very important. So I read in Josh Mittendorf's blog the news article written by Josh, and I talked to Nina. So what is this 54%? And I thought that researching like 10 minutes would be enough to I understand that this was probably something wrong and something, you know, because every time we see news articles like, wow, super rejuvenation result, and if you really go to the internet and research a bit, you in some minutes you say you perceive that this probably is not something worth it. But in this case, when I researched a bit, the opposite happened. The thing seemed to me to be more robust. Like, for example, when I learned the history of Harold Katcher, he's a scientist with his whole life he researched aging, and he was involved in the discovery of the gene of
the human breast cancer gene.
Yes, in 1994. So
in the 90s.
And then I saw that the first thing I did was to write to Aubrey de Grey, because we translated and published Aubrey's book in 2018, and we were, you know, a bit close to Aubrey. So the first thing is I wrote to Aubrey and wrote, Aubrey, did you see this? What do you think? You know, I was expecting Aubrey would reply something like, "Oh yes, this is completely wrong. You don't need to give attention to this because this, and this." So what amazed me is that Aubrey didn't reply that. He replied something ambiguous like, "Well, I don't know. First, it seems too good to be true." Like, that was the first argument of Aubrey. So that's not really an argument. And then he said, "Well, we have to see that the experiment were done by a person without a lot of supervision." Well, Aubrey had some arguments to be wary about it, but what amazed me, it wasn't like a blunt answer, like no. So this coming from Aubrey was very significant. So okay, let's dig a bit more. So one important author of the article was Rodolfo Goya, a scientist from Argentina, that I already had heard about him, but I didn't know him personally. So I contacted Rodolfo. This was 5 years ago, more than 5 years ago. And today we are very good friends with Rodolfo. We visited him in La Plata, and we met in Buenos Aires. So since in these 5 years, many things changed. But then Rodolfo explained us the experiment, and that was another thing that made us to think, wow, but this seems like something real and not some scam or marketing thing. This seems a scientific experiment. So that made us to contact Harold Katcher. Like, I think I contacted on LinkedIn, like in 2020. Then we started to talk by mail. And because since the beginning, our interest wasn't just, support, like, yes, this is true, this is rejuvenation invented. No, since the beginning, our intention was to, well, let's clarify this. Is this true? So everything we did since was to know if it's true. So we talked to Harold, and he presented us Akshay. It was his partner in the company. And then as we edited and published books, Harold in some moment said, "Oh, I am writing a book." So okay, we said, let's publish your book if you accept. Okay. So we edited his book, prepared the book, and when it was ready, we decided to go there to launch the book and to know Harold too. So unfortunately, it was during the COVID quarantine, and I have Polish nationality, so I was able to go to the US, but Nina, the consulate in São Paulo of United States wasn't issuing visas because of COVID problem. So Nina went with me to Mexico. We stayed there 15 days, and then I went to the United States to launch the book. So there I met Harold, met Akshay, and we launched the book. It was very, very interesting, the launch. And then Akshay invited us to help the company with communications consultancy. So during 1.5 years, we helped the company with this communication consultancy. But we reached the moment that we didn't see progress, and our aim is rejuvenation. I mean, my parents are 78, 79 years old. Nina's parents are old too. So we needed, progress in the research. And we thought it was better to stop with the communications consultancy. And then in May 2024, we created the Rejuvenation Science Institute, because in October 2023, finally, 3.5 years after the publishing of the article of 2020, in October 2023, they published the article in Journal Science saying what they had given to the rats.
Yeah, Harold and his team, because in the article on bioarchives, they didn't say what was exactly the thing that they injected into the rats. They said it was a plasma fraction. But then in October 2023, they explained it in this peer-reviewed article. The whole process, they explained it that this plasma fraction, the whole process of isolating the plasma fraction, Harold even says that is the exosome-rich fraction of the plasma. And so there was a process that could be, in theory, replicated. So now in this new article of October 2023, they reported 67% rejuvenation. So it was 54 before, and then Steve Horvath did new analysis because he did improvements in his epigenetic clock and things like that. And now they were reporting 67% of epigenetic rejuvenation. So like we always are looking at scientific articles in this area, in the rejuvenation field. And there are a lot of other researchers in other parts of the world that are working with exosomes. They're showing very impressive results. So this is like the line of research that we are most interested in. And so as they reported an even bigger epigenetic rejuvenation, we were like, well, someone has to reproduce this. We weren't seeing anyone trying to reproduce this experiment, this method of extraction, and try to do step by step what they did and see the results. So we were already friends with a professor here in our city. We live in a city which has a university called Unicamp, which is one of the most important universities in Latin America and in Brazil in terms of research. Sorry. And so we talked to him. He already worked with exosomes, but more in the sense of sorry, I need water. But more in the sense of understanding.
Take water.
Yeah.
More in the sense of stem cells from adipose tissue, stem cells, exosomes. So Maury worked already almost with the same thing that Harold was working. So we knew Maury from some years, but we were more inclined to other areas of research. So when we started to be interested in small cellular vesicles, exosomes, Maury said, "Oh, finally we are in the same page. So let's do something together." So fortunately, we live in this city, and we are able to make this collaboration with Unicamp, and they have a complete lab, all the equipment necessary. Besides, Maury has published it in Nature and the most influent scientific journals. So one thing that I was thinking about is that in 2020 we didn't know what they had given to the rats. So it could be anything. I mean, it was a secret, and so we were like lost. Like all we had was the trust in Steve Horvath and Harold Katcher, However, after the publication of the article in Neuroscience in October 2023, it changed, because when we knew it was exosomes from a young source, we thought, well, but there are many articles that also indicate that. And that increased our interest, because let's suppose it was a completely different technology, a complete new technology, something nobody heard about, something that Harold invented. Well, it would be different, like, well, maybe it works, but it would be the same, just trusting in Harold. But when we knew that it was small cellular vesicles from a young origin, we thought, well, but there are many indications of that. So it makes sense. So that helped us to start the institute, and we decided to make a non-profit entity, and it wasn't an easy decision. I mean, it could be a company, but the main reason to make a non-profit entity is that we think that the secrecy in the rejuvenation field is not the best policy right now for the field. I mean, the secrecy is linked to the intellectual property and patents, and is linked to the investments. I mean, people who invest need the secrecy to recover the money later. Okay, I understand. However, in the rejuvenation field, we are very in the basic science point. There is no proof of principle yet. There is no rat or mice really rejuvenated. Not just life extension. I mean, rejuvenation. To take an old mice and transform it in a young mice. Nobody did it. Did that. So,
or at least nobody that was reproduced, that was confirmed.
Yes. So maybe they did it privately in their companies. I don't know. But so we are in a point that the secrecy is counterproductive, because if you take the wrong line of research, you will waste all the money. We have many examples, like people of Google that invested in 12 years ago with a company, Calico, billions of dollars. The Amazon owner that invested billions some years ago. If you pick the wrong line of research in basic science, you can lose everything. So investors want something more, predictable, something that can return the money invested. And so there is a lot of secrecy in this field. And we thought, for example, the Harold Katcher company in 2020, they didn't reveal it, what they had given to the rat, and they took 3.5 years to say it. 40 million people die every year from age-related diseases. 40 million in 3.5 years. You have like 135 million people dead if we delay it, the arrival of rejuvenation, 3.5 years. I'm not discussing the attitude of Harold's company specifically. I'm discussing the convenience of the system of investment and intellectual property for rejuvenation science now. Once the proof of principle is done, like once we rejuvenate a rat or a mice, and everybody can do it in their labs, okay, this is how we rejuvenate a rat, even if it's a very basic process. Once the proof of principle is done, maybe it's possible to make companies to develop that. For example, when the plane was invented, to talk about something that is very close to David Wood, because he wrote the book The Abolition of Aging, and he talks about the invention of the plane. So after, Santos Dumont is a Brazilian that, so for us Brazilian, he invented the plane. For the people in the United States were the brothers, right? Okay, but when Santos Dumont fly around the Tour Eiffel in Paris, and with his plane, after that, okay, the governments and the companies, everybody went to the technology and developed it rapidly, the plane. But the plane of Santos Dumont was very basic, but he proved the principle. It wasn't a very big company or very big, even the brothers Wright, they weren't a big company, a big capitalist or a big government. No, no, no. The proof of principle is different from the technological development. So what we want to do is to prove the principle. And we are not invented anything. We just saw the article of Harold, and we thought that the system of incentives wasn't prepared to immediately repeat that, to verify if it was true. So we were like two people in Brazil. But we thought, well, we have to do something, because I think that eventually this will be proven in some way. But this is urgent. This should be done now. And so we didn't have another option than to make this non-profit institute, and to make the collaboration with Unicamp, with Professor Mori. And last year, we did the first part of the experiment. We already produced the plasma fraction of Harold Katcher in the university here last year, and we injected in young rats, because that is a story. I believe Alfred Gray will be okay that I tell you. I hope, but that's nothing very important. But we had a meeting with Alfred de Grey, Didier Cornell from Hillis, and Professor Maury, me and Nina last year, and at the beginning of 2024. And Albright said, I believe that in the moment you inject the preparation from young pigs in rats, they will die from anaphylactic shock. And we said,
also because it is a very big amount. Harold reports injecting a very high amount of this plasma fraction.
Yeah, an extremely high amount. It's ridiculous high amount of pig's protein in rats. So Albright said, I believe they will probably die instantly of anaphylactic shock. So instead of doing the whole experiment and raise all the money and take all the work, why don't you do a small experiment and injecting young rats, like 3 young rats, just to see if they die instantly? And then you don't need to make the whole experiment. You can just report that the rats died. And Didier from Hillis thought it was a good idea. He funded an experiment with $5,000 because it was a small experiment. And we did it. So I went to a farm here, 3 hours from here, with a veterinary. We collect the blood of the young pig. We went to the university lab together with Dr. Mori and a scientist that works,
postdoctoral researcher.
A postdoctoral researcher. We did the isolation, then we injected in the rats, and there was nothing wrong. Like no signs of toxicity, acute toxicity, no signs of acute immunogenicity. The rats continued to eat,
to gain
weight.
We did also histological analysis to see the organs, and they were fine. Like nothing in this acute experiment showed any signs of toxicity.
And when we showed the results to Albright, he was very, surprised and happy. Wow, now I'm more confident in this. And then we decided to proceed to the whole experiment, and we set this together with the university, this budget of $75,000, and started to raise the resources. Then we published the article in the European Journal of Biological Research, reporting this experiment of 2024. And up to now, we raised around $55,000 is around 73% of the total, and it lacks $20,000 yet. And on May, we will go to the farm to collect the blood of the pigs. I mean, 3 days ago, the farmer informed us that 10 pigs were born recently. So
because we need the blood of around 10 pigs, because in this experiment that we will start in May of 2026, we will actually have two treated groups. We'll have 25 months old rats, which is the same age that Harold used, but we also have 7 months old rats, because Professor Morris suggested that we could also test if the plasma fraction
could keep young rats young.
Sorry, I forgot.
And not just rejuvenate rats,
Yeah.
but keep young rats young too.
Yeah, so we'll have 10 rats that are 7 months old, 10 rats that are 25 months old. So also it's a bigger amount of rats than Harold used. I think he used six rats. And also have control for both, so 10 young and 10 old. And so we need around the blood of around 10 pigs for that. And yeah, so we raised it, as Nicolás said, more than 70% of the budget. With this money, we can do the experiment, but not all of the measurements, because we want to do grip strength, memory tests, that is something that Harold did, and also blood markers and epigenetic age. If we don't have like the full amount, we will have to keep the blood of the animals and do these blood markers and epigenetic age later when we have more money. But this funding came, a lot of this 70% that we have, $53,000, something like that. So a lot of it came from donotage.org.
Is $55,000 from Do Not Age?
No, 25.
$25,000, yes.
Yeah, it came from donotage.org, and then $10,000 from Hillis, the NGO from Belgium, led by Didier. And also a lot of donations, big and small, like $1,000 donations and $3 donations that we have so far. So yeah, and we are trying to raise this money to do the whole experiment. And also, ideally, if we see signs of rejuvenation, or if we see that the rats are not aging, for example, we also want to keep these animals alive and keep giving them the plasma fraction to do a longevity study to see how long they will live. So, for that we need a different budget. So this 75 is basically to reproduce Harold, which is a 5 months experiment in terms of when the rats start getting the injections.
Yeah, we up to now we had 39 different donors, and the fact is that 95% came from 3 or 4 donors. The others 35 donors gave smaller amount. We are very happy with any donations. Like recently, a person from Chile started donating $3 per month, and we think our vision for the future is that, like, thousands of people giving just a bit. But to be honest, we are so close to the beginning of the experiment that I suppose that donations a bit bigger will be important. Not in the long term. In the long term, we want small amounts from many people. Well, regarding Harold Katcher, just a bit of word about it. Harold is a very, very special person. I call him every month to see how is his health. He's 81. He will be 82 years old in a few months, and I'm a bit worried about his health, because there are people with 81 that are very well, but it's not the case of Harold. He's doing his best to advance with the research in the way he can, but Harold chose a path uh, many years ago, 10 years ago. He chose the path, a different path, economically speaking. He chose the path of the intellectual property and patents and companies, investment and profit. He that is his line, so we are trying another path. We just want to know if it's true. And his company is trying the patent, is trying to get the patent since 5 years ago. And as patents are, it's a process that can take decades. But as I told you, every year 40 million people die from age-related diseases. So if you wait 20 years to the patent, but we are just reproducing an experiment, publish it in a peer-reviewed scientific journal. So I believe this is in line with all requirements, and it makes sense to verify if so important experiment could be reproduced. So I talk to Harold, every month, to check if he's all right, and well, I hope we can reproduce, rejuvenate Harold too, right?
Yes, absolutely. And I hope that advances in human rejuvenation will come in time for those alive today, especially many scientists and transhumanist advocates who are currently in their 60s, 70s, 80s. And I think there is a realistic chance, if Aubrey de Grey is correct in his probabilistic time frame, and his current estimate is of a 50% probability of longevity escape velocity arriving 11 to 15 years from now, so in the late 2030s, that would be a sufficient amount of time to help those who are currently in those decades of life and who are in good health. So if they're able to at least maintain decent health until then perhaps rejuvenation biotechnology will help them. And certainly that is my hope, and that is the hope of essentially everyone in our movement. To our audience, I wanted to highlight several links once again. First of all, here is the website of the Rejuvenation Science Institute. If you're an English speaker, this link will take you to the English language version. And also here is a link to make a donation to the Rejuvenation Science Institute. As mentioned, the majority of the funding thus far has come from several large donations, such as from donotage.org and HEAL. So I've highlighted their websites for you to check out, but small donations are also welcomed. And small may be a relative description. So based on your resources, I donated $100 recently, and for some people that's a small amount, For some people, that may be larger than they could afford. And if you can afford $10 or $3, I think that would be welcome also. So Mike Lazine wonders, would smaller donations be more ideal, because he thinks if you get large donations from rich people, they might be transactional like Trump and want a return of so-called favors. But of course, at this stage, with this scale of funding, we're not talking about funders like Trump. We're talking about people who are aligned with the mission, people like Didier Cornell, for instance, who has been known to make donations of several $1,000s, sometimes tens of thousands of dollars, to these kinds of experiments that may only require money on that scale. But I think to Didier, something that is important is the experiment tests something that hasn't been tested very often, that has been ignored or bypassed by a lot of studies for various reasons. And one aspect that I think is noteworthy is the potential for a longevity study here, because all too many experiments that try to show rejuvenation, they essentially have an arbitrary cutoff point, like after 60 months or something like that, or even a shorter time period, after a year, they just say, okay, this is it. We're stopping the experiment, and the animals get euthanized, even though that treatment could have prolonged their lives. So I think it's really important to do that longevity study. And I know you have some additional budgetary requirements that would be needed, but this was what was remarkable about Harold Katcher's experiment. It produced Sima the rat, and that made headlines. And I think that's important for advancing people's understanding of what is possible in terms of rejuvenation. But I also wanted to reinforce your point about nonprofits and transparency of research. So the way you've structured this would essentially avoid the need to keep secret what you're doing and what you're administering to the rats. So it would all be accessible to other scientists who would want to do similar experiments or variations on those experiments. And that could also leverage the ability to make advances in the field, because they could have additional resources that they would do their own experiments with. Now, for our viewers, I just wanted to confirm the size of the experiment. So you would have 10 young rats that you mentioned, around 7 months old, and 10 old rats around 25 months old. And then you would have 10 young controls and 10 old controls. So 40 rats in total for the experiment. Is that correct?
Yeah, that's correct.
Okay.
Yeah, we'll inject the preparation at the beginning of the experiment and 3 months later. And 2 months after the second injection, we'll measure again the epigenetic age.
Yeah, because we another difference from Harold's work is that we want to measure the epigenetic age before the injections and after. So this 5 months after the first injections, which is something that he didn't do, but he did like in several tissues, because they killed the rats, they euthanized the rats. But we want to keep the rats alive, so we will only do the blood. So we'll do the blood before and after.
Yes, that's very good, and I think very promising for you to take that approach. And one more link that I will share before turning the floor over to David Wood is a link to last week's stream that we did in collaboration with Transvision Madrid. We have done 3 streams about the recent Transvision Madrid summit. And this particular stream that we showed last week included remarks by Dr. Steve Horvath on epigenetic clocks. So please check that out if you haven't seen that yet, because you will see Steve Horvath's work highlighted, and he would be a great collaborator for any sort of study. I'm glad that you have him on board. So David, please go ahead with your remarks.
Well, first thing to say is this is an extremely interesting set of ideas, and I really hope this project can go ahead quickly. It is novel, and with the understanding that where the plasma is coming from, it suddenly makes it extremely interesting. My first question is to Nicolás and Nina. There is a kind of difference in view between what Aubrey has been suggesting and what Harold has been suggesting. Aubrey has said, well, aging is multiple types of damage. You know, there is damage in the mitochondria, there is damage in DNA, there's damage in the extracellular matrix, and so on and so on. Therefore, we're going to need multiple different approaches. We can call this a plural approach. Whereas Harold Katcher has said, well, actually, there's one or two things, maybe just one thing that controls everything else. And in Harold's view, it's the signaling between cells. You know, the cells are sending messages to each other the whole time. And if the cells think, well, this body's getting old, it puts them in a different mode. Whereas if you could change the signaling, as he hopes to do, or maybe has done with the exosomes, then that by itself will be sufficient to rejuvenate. And we wouldn't need multiple types of damage intervention. So where do you stand on that? Bearing in mind that after all, Sima did die in due course, despite getting some rejuvenation treatments. Maybe Sima would have needed more often treatments, or maybe these treatments only repaired some of their types of damage and there will be yet other treatments necessary for the other aspects of aging.
Yeah, so yeah, you are correct in terms of how Harold views aging. He basically sees it as a signaling process that is controlled epigenetically. So the idea is that the signals somehow change the epigenetic of the cell, and then the cell by itself could use its own mechanisms to repair itself. So our opinion is based not only on Harold, but like there was a paper by a group from China in 2024, I think, that they injected young plasma from humans and from rats, or from mice, I think, into mice. And not the plasma, sorry. They extracted from the plasma the exosomes, the small extracellular vesicles. And they measured a lot of things that Aubrey mentions in his book, for example, lipofuscin, I think that's how it's called, that is like a intracellular kind of garbage that accumulates inside the lysosomes that can't be degraded. And so they measured that, and the old rats I think it was mice, the old mice that received the treatment, they had less lipofuscin. So it was like an example that maybe Harold is correct in the sense that the cell maybe has the mechanisms but to repair these damages, but they are just not, the cells are not employing these mechanisms because they receive a signal that says, no, you need to be old, you need to be degrading, you know. So I think in general we are more aligned with this idea of the signaling, but we're trying to check if this experiment is true. So it's not like we have like conviction, you know.
And in this experiment of the Chinese group published in Nature, I think it was in April of 2024, they also measured the ages that
ages is advanced glycation end products. So it's formed in the extracellular matrix. Yeah,
is one of the 7 basic damages that Aubrey describes about aging. And the most interesting is that it's something in the extracellular matrix. It's outside the cells. So apparently, according to this Chinese experiment, the young exosomes, interspecies, because it's human to mice, were able to rejuvenate not only lipofuscin, that is another Aubrey degrades the basic damage inside the cell, but also something outside the cell, the ages. So it's interesting your question, David, because Aubrey and Harold are our 2 references in this field. I mean, and I remember I was talking to Aubrey by email in 2020 when Harold published the article. And I was having a conversation by email with Aubrey because there was something that I didn't, accepted, in his Aubrey's theory, that it was like, for me in that time, I saw that Aubrey had described it like the 7 basic damage. They were like 7 basic diseases, 7 basic problems that our genes can't solve for evolutionary reasons. But Aubrey was able to, instead of talking of the diseases downstream, like Alzheimer, of atherosclerosis, he was able to go upstream until the molecular cellular level to describe what are the basic diseases. But they're, let's say, diseases, they're problems our genes aren't capable to solve. Okay, and Aubrey said, this is aging. We don't have the genes to solve them. So when I saw Harold's article, I thought, hey, maybe there is something upstream to the 7 damage accumulation process that Aubrey described, something that regulates them. And that's what I think about Harold's theory. Harold described a mechanism upstream to Aubrey damage processes. I mean, it's something that by signaling tells to the cells okay, your damage repair level should be that of let's say 40 years old person, or your energy production should be this, like a 70 years old person. So and then if you don't have enough damage repair, of course the 7 kind of damage will accumulate. So what Harold says is something upstream what Aubrey says. And Aubrey, I already talked to Aubrey about that, and Aubrey says, man, I don't care how this damage is repaired. If you find a way to repair the damage using an upstream process, that's fine to me. So that's our position. I think they are both very good biologists, and we want to use, this potency, and not to have like discussions and on opposition. It's just we are all in the same page here, I think, or almost in the same page. And sorry, Nina went to help Aurora a bit, but she will be here in a minute.
My next question is about E5. How confident are you that you understand now what's in E5? Well, you can actually replicate the experiment by yourself.
Yeah, E5 is the name Yuvan, the company, gave to the preparation. And the preparation, according to the article in 2023 in Neuroscience, is extracellular particles. It's not just small cellular vesicles, it's not just exosomes. And Professor Mori told us that the Society of Small Cellular Vesicles recommends that when you have a preparation that includes not only small cellular vesicles, you have to call it extracellular particles. So that's why we call it extracellular particles from the plasma of young pigs. So there are small cellular vesicles, exosomes there, because Harold made this test in this seminal experiment. But even Harold, there is an interview of Harold in which he said that maybe there are another particles there, like exomeres or supermeres, some particles that can be part of the effect. So maybe it's not just exosomes, this preparation. The fact is that this is a posterior step. Like, first we have to be sure that this rejuvenate, because if this rejuvenates in 67%, the rats and the memory and the grip strength and everything, and this, can be replicated and established, okay, we discovered, okay, Harold Katcher article is right, the humanity knows how to rejuvenate mammals. Then there will be another step, like, for example, to know in this preparation what exactly are the signal, which exactly are the signals, the signaling of aging. It will probably need some kind of repeated experiments or the use of artificial intelligence to decipher the code. I personally believe that there will be a code. The cells communicate using a code, and we have to decipher this code to know how the cells communicate, and use this code to tell the cells, okay, you have to have the phenotype of a 25 years old person and not a 70 years old person. This will be a posterior process. And it's funny that 5 years ago, when I first saw the article of Harold in Josh Mitteldorf's blog, the first thing I thought was that it was some kind of code there. Like, for example, the Yamanaka genes are just an example. They are able to rejuvenate a cell, not an organism yet, but a cell. Okay, so, but maybe there are other signals besides the Yamanaka genes. Maybe there are thousands of signals a part of, the 4 Yamanaka genes. So I remember I talked to Michael Ray from the Science Research Foundation about that in 2020. Michael, there should be some kind of code, So I have this idea since then, even before I knew that it was small cellular vesicles. So to answer your question, David, first we have to know if it works, and then it doesn't need to be us to know who it is in this preparation exactly, because David asked what is it in the preparation precisely. So I said, first, let's see if it works.
Yeah, Harold, well, first of all, sorry, I had to go change a diaper. Our daughter needed us. But yeah, so Harold didn't do like a full characterization of the plasma fraction. He just did a Western blot that confirmed that there are small extracellular vesicles present in the plasma fraction. So this is something that we want to do as well during the longevity experiment. If we have the funds, we also want to do a better characterization. So we don't know exactly what it is. We know in general that there are small extracellular particles, because we can't say for sure that it's only exosomes or only small extracellular vesicles. But yeah, this is something that is also important to see what it is exactly.
Yeah, because we don't want to use pigs or cattle to rejuvenate 8 billion people. I mean, it's impossible. It's not ethical. It's terrible in all senses. We have to understand what it is and what is the code. Is the main point for me, to synthesize that in some way, and don't need to use animals. That is just the proof of principle. It's not.
So why pigs currently? Is that just for convenience, that it's easy to get the blood from young pigs as opposed to blood from other animals?
It's basically because we want to do as close as possible as what Harold did. Because
But why did Harold pick pigs?
He said that it is because pigs are relatively similar with humans, I mean, if you have to choose an animal, pigs are relatively, the anatomy is similar. So
yeah, that there are some things from pigs that we use, like for heart valves and things like that we use that are from pigs. So I think it was something like that.
yeah, and the pigs are, already eaten. Like 1 billion pigs are eaten every year in the world, and the blood is usually wasted. So that, helps too, I believe.
Thanks.
Yes, thank you for those answers, and also for those who are interested more in the ideas of Michael Ray, since he was mentioned. He gave a comprehensive presentation for our virtual Enlightenment Salon on August 6th, 2023, as to his assessment of the state of progress in longevity research and in each of the 7 SENS strains of damage repair. But it is a very intriguing idea that there may be something that could be affected upstream of multiple types of age-related damage that could lead to a convenient, concise damage repair approach for several of these types at the very least. John H. has an interesting comment. All biological roads lead to oldness. Find that one thing and the rest falls into place. That idea appeals to me very much. It's spare and elegant. And he later stated he likes the way Nicolás and Harold are thinking. So thank you very much for those comments, John. And here is a link to the paper by Harold Katcher and his team. This was published in December of 2024 in the Aging Cell magazine. It's entitled E5 Treatment Showing Improved Health Span and Lifespan in Old Sprague-Dawley Rats. So take a look at that paper if you are interested in additional details about the study. And then we have a question from Mike Lazine. He wonders, wouldn't we also have issues of the human body's T cells or white blood cells attacking animal plasma or blood? How do we get around that? So if you're injecting rats with pig plasma, would there be an adverse immune reaction? Or have you found in your safety study essentially that this doesn't happen?
Just one thing, the article you mentioned by Harold is the second, and is another one of 2024. Is the Sima's article. The article in which they describe the first experiment is from 2023 of in Neuroscience. Okay, just a comment.
Yeah, about this is definitely something that needs to be ensured that this plasma fraction is safe. So in our experiment that we did with young Sprague-Dawley rats, we injected a huge amount of this plasma fraction into the animals. And in terms of an acute experiment, so it's not like long-term, we didn't see signs of toxicity or any signs of immunogenicity. So didn't see signs of the blood cell, the white blood cells trying to attack the plasma fraction. There are also a lot of experiments from other groups that they inject exosomes, small extracellular vesicles from human blood into rats, and they also seem to be fine. Like there is no immune reaction. So they seem that in a lot of articles, other researchers say that they have low immunogenicity, the small extracellular vesicles. But of course, if this research is to carry on, because we're first just trying to reproduce it, but then we will, if it is reproducible, if the rats rejuvenate, if they live longer in the longevity experiment, we will have to do a full safety experiment. So this is something also that we're thinking about that is much more expensive. but that is something that needs to be done as well. And after that, this is in animals. So a full safety study, doing everything according to like FDA and the European Agency for
EMA.
yeah.
EMA.
I don't remember what exactly is.
The European Medicines Agency, correct?
Yes, that's right.
Yeah.
So yeah, so we want to do that as well if we are successful. And this is done first in rats as well, in rodents, and then usually they test in another animal species, so like primates, non-human primates, before testing humans. But here in Brazil, I don't know if it's the case in other countries, but there is like a group of patients that they call compassive patients, that are patients that like they are dying, they tried everything that they could do, they have no other treatment option. If you have something that is proven to be safe only on rats, you could give it to them, you know. So in theory, if this is effective and this is proven to be safe only in rats, even if only in rats, we could try to do an experiment in humans. But in this case of people who are already dying, they have no other choice, no other treatment, they exhausted all their options. So this is something as well that we are thinking, but this is much ahead.
Yeah, but Michael's question is extremely important, but that was the line of thinking of Aubrey de Grey. I mean, that's why we injected the pigs preparation in rats, and the rats didn't have any problem. And as Dina said, there are many studies. They are in the bibliography of our article published the last year. Several articles showing interspecies injection of exosome, small cellular vesicles, and without any immunogenicity. So, you know, Michael, that's it's a question that should be answered by experiments, but not just by experiments. You know, in the book of Harold Katcher, The Illusion of Knowledge, he made an extensive rationale, an explanation since the beginning of life, 3 billion ago, to explain why the mechanism of age determination should be shared a lot by the living organisms, and that would explain why mammals would have the same mechanism. Because, you know, Harold explains how even unicellular beings 3 billion years ago could already use signaling to regulate aging since aging began. Because Harold say that aging began with sexual reproduction, because, you know, bacteria don't age. I mean, they reproduce asexually, and so they don't have aging. They can die, but they don't have this process that sexually reproducing beings have, aging. So as Harold explained in his book, he's a great biology. I mean, that's incredible in Harold. He's a biologist. He's not an entrepreneur. He's not showman. He's not other things. He's a biologist, and he explains how this code could be very ancient, the code of regulating aging. So maybe mammals share this code, and the experiments show that. The experiments are showing that interspecies injections of exosomes are not immunogenic. So we will continue to do those experiments. And as Nina said, if this experiment works, we intend already in 2027, after this experiment, to start the safety experiment here in Brazil, in Santa Catarina. There is a facility that is probably the only facility of Brazil that can test rodents, safety rodents, according with Brazilian authorities' requirement. Until 2014,
and not only Brazilian requirements, FDA, EMA, they follow the same guidelines.
Yeah, also because Brazilian authorities follows FDA and EMA guidelines.
They copy paste.
Yeah, no, it's not copy paste. It's more. They say in the law, in the Brazilian law, it should be the requirements of the FDA and EMA. So until 2014, there wasn't any facility in Brazil in which you could test something in rodents according to the Brazilian authorities' requirement. So in 2014, it was created in the state of Santa Catarina, a facility called CIEM, which tests according to good laboratory practice. So this is it costs like 10 times more than this experiment we'll do next year, but is approved by not only, as Nina said, not only Brazilian authorities, but follows the requirements of the international good laboratory practice. So after that experiment in 2027, if everything goes well, we could try it in compulsive patients, because there is no lack of people dying at any minute. But I hope, in fact, that other scientists and laboratories in the world could do the same. And if we test safety in this internationally approved facility in Santa Catarina, people in other countries could use that test of safety to test in compulsive patients, who don't have any option in any country. That is what we expect. It's not just Brazil. It's not just us. We want to make everything public in order people around the world do it in their countries, in their legislation.
Yes, indeed. Thank you for that thorough answer. And I will also post a link to the book that you mentioned by Harold Katcher, which is called The Illusion of Knowledge, the paradigm shift in aging research that shows the way to human rejuvenation. It can be found on Amazon, and here is the link. Definitely a good read to get some insight in how Harold Katcher views aging and the approaches that could be pursued to combat it. So thank you very much. And we have an interesting question from Art Ramon Garcia, and he wonders, can it be assumed that this fractional plasma only benefits a mammal if it comes from a longer-lived mammal donor? And where this comes from is the observation that pigs are longer lived than rats, and the pig donors' plasma were used to rejuvenate the rats. So if something analogous works in humans, would the plasma have to come from mammals who are longer lived than humans, such as certain whales, for instance, or even a Greenland shark, though it would be hard to get anything from a shark? Would it have to come from some creature that's longer lived than humans?
Yeah, that's a very interesting question. Sharks are not mammals.
Yes. No, they're not.
But yeah, so I think that the idea from Harold is that no, that it doesn't needs to be long-lived, longer with a longer lifespan. The another reason that he used pigs is that he wanted to give a very high concentration of this aging signaling. So if he would take from a rat, from the blood of a rat, he would need a lot of rats. So the idea is that to take a large animal and then you concentrate it and then you inject it. And I think he was thinking about like 4 times the amount that the animal already has in their body. So if they have like a signaling that says that he needs to be old, he takes like from a bigger animal the signaling, because the animal is younger, the signaling that says it needs to be young, and concentrate it in an amount that is like 4 times the signaling that the old animal would have to overcome the natural signaling from the animal. I think it wasn't based on like pigs being longer lived than rats. Basically, I think his idea is a bit similar to what also Dr. Steve Horvath said, that like his epigenetic clock measures like what percentage of your lifespan you have lived, you know. So it works for the same epigenetic clock works for a lot of different animals, a lot of different mammals. I think he made a epigenetic clock that works for all placental mammals, based on this idea that how much of your lifespan you have lived, like 70% or 50%. So I think Harold has the same idea in the sense that if a pig lives, I don't know, 12 years, I don't know how long a pig lives, but if you take his blood when he had lived, I don't know, 20% of his lifespan, then it's similar to the signaling that he has in the blood is similar to a signaling that a human would have at 20% of their lifespan. I think it's basically that.
Yeah, I mean, in the rationale of Harold, the signaling indicates to the cells they should behave in a young manner, like a young phenotype. So if the pig is young, it would mean the cells are receiving these signals telling the cells to behave young. So in that explanation, if you inject those signals in a human which lives more time than a pig, but the signals will indicate that the cells should behave in a young manner, corresponding to, as Nina said, the percentage of the live that species live. So is like signaling of youth. Youth for a rat can be 7 months. For a human can be 20 years. For a whale can be 60 years old. But this youth, because is a determinate age phenotype, that is a determinate degree of efficiency of repair mechanisms and energy production.
Very interesting. Thank you very much. And Art Ramon follows up with another question. So could humans get this plasma from the calves of cattle, for example, because they're young creatures?
Yeah, we think so in theory. Like that, Harold chose the age of the pigs because it's the blood of a pig that is around 6 or 7 months old. So the idea is that is a pig that already is post puberty, but is very young. So I don't know, like 20 years old if he was a human, something like that. So I don't know the age of the calves, how old they will have to be something equivalent, that they already are post-pubescent.
Puberty.
Yeah, post-puberty. But but I think in theory you could use other mammals.
Yes, indeed. We are waiting for people to do it with other species. I mean, it will be very interesting.
Yes, so it's interesting too, the question of pig lifespans arose. Here's the Wikipedia entry on pigs, which suggests that the maximum recorded lifespan for a pig is actually 27 years, and on average, in captivity, if they are not eaten, pigs can live for 15 to 20 years. So actually, that is quite a bit longer than I think most people would expect of pigs, but some pigs can outlive dogs or even cats, though cats with good care in captivity can live for 20 plus years, and the longest lived cat was actually 38 years old. But this is a fascinating discussion as well, because if these kinds of exosome treatments from animal plasma are successful in rats and this can be replicated, the logical follow-up questions that a lot of people will have will be along these lines of what can work in humans and how can this be scaled up. So thank you for your responses there. And I will also highlight a comment that was made by David Wood, because he had Harold Katcher as a guest in the London Futurists webinar 4 years ago, entitled A Paradigm Shift in Aging Research. It has over 4,400 views right now, so please go ahead and watch it after the salon. And David, I know you have another question, so please proceed.
Yes, so my question's slightly philosophical, but and I'm not trying to say we shouldn't do experiments. I absolutely agree that doing the experiments is a scientific approach. We should do the experiments with an open mind. But I want to come back to this possibility that there can be a single thing that will reverse aging rather than multiple things. And by the way, as I've been listening to you, I've also been talking to Aubrey. Not Aubrey the person, but Aubrey the bot. That's AUBR.ai. I've been having a fascinating conversation with the bot about Harold Katcher's experiments. And it matches what you said, that Aubrey thinks the experiments are indeed worth replicating and going into. The bot does think, however, that there's probably more than one component in the E5 that's important. There's probably several different things that needs to be put in. So even if it's just one treatment, there's probably several things going on. But my question is this, because I've heard Aubrey, the real human one, come up with this objection several times. He says that if there was a single thing that could make a difference between bodies aging and not, then evolution would have found it. And there would have been some organisms that would have said, you know what, I'm going to not do this decline. I'm going to keep my signaling youthful. And these individuals would therefore live longer. And naturally, they would pass their new genetics down to subsequent generations. So that's one argument why there has to be multiple things that need to be solved to solve aging. Otherwise, if it was just a single thing, we'd see long-lived species emerging. And I know Harold objects to this because he says, no, you shouldn't think about it as evolution of the individuals. You have to think about it as evolution of the species. It's good for the species if there's plenty of variety and individuals die off, so that there can be variety in subsequent generations. But the counter argument to that is always, it's the objection to group selection, which is that individuals who don't follow that general template will actually tend to live longer and their genes will tend to multiply. So I don't know if you've any thought on that, or maybe you just want to stick with the experiments and let's figure out whether this works or not.
Yeah, so I think you are correct in what Harold says about that. He talks about it in his book, not only about the variability, but also like if you have, for example, a wolf that is 50 years old, he's strong, he doesn't age or age very little, and he knows everything like an old wolf knows, like that there will be no competition for the young. So yeah, it's this idea of group selection, not the individual selection. That also in terms of ecological niche, like not only inside the group, but how this animal relates to his ecological niche, you know. But like personally, like I'm not a biologist, I can't answer that very well. Uh, we basically want to see Harold. I think he mentioned this. I think he mentioned in some interview that he thinks in his plasma fraction is not only the exosomes or the small extracellular vesicles that are doing something. He thinks other small extracellular particles there might also be contributing to the effect, like exomeres, supermeres, things like that are in the blood. We also talked because a lot of research in small extracellular vesicles, they try to find like what microRNA inside the small extracellular vesicles is responsible for the result, for example. And we talked to a researcher from Spain. She also works with this, and we asked her about that of the microRNAs, and she talked that she thinks the whole small extracellular vesicle, like even the proteins in the membrane and everything might be contributing to the effects. It's not just one thing inside, you know. So in general, I think Harold, and in theory we agree that it is like a combination of a lot of things. It's like several types of things inside the sorry.
Yes.
Our
Nina will go with Aurora. I will continue. David, you did very well. The narration of the argumentation between Aubrey and Harold, because when you said what Aubrey said, I would say what Harold said, but you said first, and then you said Aubrey's answer. But the example Nina gave, it's good. I mean, imagine a wolf of that randomly has a mutation that he can emit the signal of youth forever. So the wolf is 300 years old, and if he has like descendants, little wolf, they won't stand a chance against this wolf of 300 years old and in his perfect physical shape. So it's a wolf that knows everything that a wolf can know, and very strong. So evolutionarily, okay, this wolf won't evolution won't advance in that case. But as Harold says that precisely that a fixed and a limited lifespan for the species is necessary to the velocity of evolution. Of course, that is an important question, a theoretical question, but I suspect that experiments and the practice will answer that. I mean, once we have some proven techniques and experiments, we could also better this question. But it's indeed a very good question. But there is another thing in Harold's vision of aging, a vision that goes to billions of years ago. This mechanism is too old, and this aging mechanism is too old, and maybe that influences this evolutionary thinking. But I agree, it's a very interesting question, and I think it will be answered by experiments.
Yes, thank you for that answer. And we have an interesting question from Super Fin Guy. He wonders, do all animals that reproduce sexually age? Because we know of several species of animals that are negligibly senescent, so their rate of aging is a lot slower than even that of humans. For instance, certain species of tortoises, or the whales that we mentioned, or the Greenland sharks, or even naked mole rats. Now, we don't know exactly how long the naked mole rats can live, but in a lab setting, they've been observed to live for as long as 40 years, and one wouldn't expect animals of their size to have such longevity, but it does seem like tissue samples from naked mole rats who are older do not show that much more senescence as compared to ones who are younger. And all of these animals that I mentioned do reproduce sexually. So is it necessarily the case that sexual reproduction requires aging, or is it just more of a probabilistic observation that organisms that have that mode of reproduction will tend to age because of this group selection idea that you mentioned?
The question is, wasn't here. The question is if all sexually reproducing animals age, for example, naked mole rats. Well,
like we are not like the people who studied biology and did all these theories. We're trying to see if this kind of rejuvenation therapies, rejuvenation research is reproducible, is real science. Like you mentioned it in the beginning, like real science is someone needs to reproduce it. So I don't know if we are the best people to answer that. Like we read a lot about it, but
yeah,
basically that's an argument that Harold makes in his book, that aging began with sexual reproduction, even in unicellular organisms that have this kind of reproduction, because they do like two organisms do a conjunction, something like that. And but yeah, we need to do research, like more people.
Yeah, but the one thing, the animals you mentioned, any of them live forever. I mean, they can age at a slow pace, but there is no animal that live forever. Even the naked mole rats. There was recently a paper in which Steve Horvath participated. I believe that they measured the epigenetic age of the naked mole rat, and it advances. They age epigenetically, and they die too. They live a lot of time, but there are no immortal sexually reproducing animals. The hydra, it doesn't age, but it reproduces asexually, as far as I know. So we have to distinguish the animals that age very slowly, that biologically immortal animals. This should be proven. I mean, I'm not sure if even the naked mole rats, if they are really immortals. I mean, I don't know if you have some information that, but there is no proven.
I think in English they call it the biologically immortal jellyfish, not hydra. It's the Turritopsis.
No. This is another one. She's talking about the Turritopsis. Okay. It's another case. It's the jellyfish that turns into a very undeveloped form, and then in a very developed form. Then again so they can live forever. doing that, the turritopsis, but it's not the case of Hydra. The Hydra is another animal, but it doesn't reproduce sexually.
Yes, interesting. Thank you for that response. And it seems that there could be creatures that reproduce sexually that age a lot slower than most animals, but they still age at a certain rate. And that seems to be, at present, a consequence of that mode of reproduction, but hopefully rejuvenation treatments can do something about it. Now, the next question has two parts, a general part and a specific part. The general part comes from Daniel Tweed, and he wonders, is there enough information and research sharing going on amongst various lifespan researchers these days? In your opinion, how would you comment on whether or not there's enough information sharing? And then the specific part of the question is on information sharing from Harold Katcher. So you mentioned it took about 3.5 years from the time that these treatments were administered to the rats to the time that his team disclosed what substance was administered. So do you know what ultimately led Harold Katcher and his team to make that disclosure? They kept that information secret for 3.5 years, but then they decided, okay, it's time to share it with the world. What happened to lead them to make that decision at that time, do you think?
Yeah, I think they were waiting for the patent to be like officially processed. Like they didn't. It's not that they got the patent, but they submitted everything, and then there was like some time.
1.5 years
Yeah,
that you have to wait
for the patent office of the United States to make a document just to guarantee that it's theirs, even if the patent in the end is not granted. I think it was that they were waiting for that.
Theoretically, patents should be made in a way that a skilled in the art person could read the patent and make the procedure. So theoretically, when the patent request was published, there was no secret anymore. So I think this process of because the patenting process is not just to request the patent, then the U.S. Office patent they are all public documents. You can follow the patenting process of anything on internet. It's all public information. So there is a process in which the U.S. patent offices say, oh, okay, but I think this, and this, and the patenting requesting entity says, okay, but I mean, there is a dialogue. And when, this article was published in October 2023, the patent request was already published. So I think that was the essential point, but I don't know.
Yeah, it's not something that we know personally. We are guessing, but like in general, like Harold doesn't disclose a lot. He's very secretive. He's worried even like with hiring people to work for him because they will know the secrets, things like that. So he's not someone that is very open to do collaborations, he doesn't tell us much or anything, I don't know. But in terms of how is the collaboration in general with scientists in this area, what I see personally is that people who work in academia are more open to disclose everything because basically the research is already paid, right? Like there was a grant from a government or something like that, and so they can disclose. Usually the most closed ones are people related to companies. Even there are academic researchers that are also involved in companies, but in general they're more open to the academic researchers are more open to collaboration and to disclose information?
Yeah, but of course, I mean, the answer is that there is not too much information sharing. I mean, some people say there are a lot of information sharing, but I think the model in the state of granting, in the universities, many times the professor asks the students not to comment with, other people what they are doing because somebody can copy. So it's not just a patenting or intellectual property thing. It's the way the academic grants work as well. So and the intellectual property system also incentivize this secrecy, and we are trying to do differently because people say it can't be done. When you criticize the secrecy, they say, okay, but there is no way to do it differently. You can't attract enough resources if you don't promise to try to get the money back, which is what happens with investment. I mean, people put a lot of money and they are promised that the money, they will try to get the money back. And for that, you need some system. I mean, if you discover something, you have to ask the state to forbid everybody to use it for 20 years. So, okay, it's a system. Many good things were done and discovered and implemented with that system. But also, the nonprofit system also invented many things in the history of science. So there are examples of both things. And I think nowadays in rejuvenation science, because we are in the basic science stage, because we are in the internet age. I mean, today, the cost of information sharing and is practically zero with internet. If you put an information on the internet, it can be distributed globally at almost zero cost for the people who share the information. So it's not the same to talk about intellectual property today and 50 years ago. Today, if we don't share something, we are wasting this terribly useful machine called internet. So I think we are trying to do differently. We are trying to do with a system that is prepaid. I mean, people who contribute financially is not promised that the money will come back. We promise to try to discover things that can help them to rejuvenate, or that their relatives are able to live and not die. We can promise to look for health and discover that will save lives. That is what we can promise. We won't promise to try to have the money back, because if we do that, we have to make a lot of secrets, and I think at this stage in which we have to prove the principle, we need the help of everybody. We need the help of other labs, other laboratories around the world, scientists. So we can't keep secrets. And regarding Harold, it's like Nina said, and like I said, 10 years ago, Harold made the decision of this path. I don't know if it was his decision. It was the way he was able to do it. He found out Akshay, and Akshay proposed this path of intellectual property, and Harold follows it. He's very secretive, that. He was interviewed by many of you several times, and he's very secretive, and he's very sincere about that. It's very sad too. I talk to him every month. I call him to see how how is his health, his family, And he told me, I would like to be able to, to share things and to say what I'm thinking about everything, but I can't. And we have to respect that. It's sad, but instead of criticizing him, we have to, okay, you have the right to do it. He have a particular situation, you can't demand so much of somebody. He had already did, he already did a lot, let's let Harold, happy, he deserves to have, tranquility. He has a beautiful family, and we have to do the things now. If the patent is approved someday, okay, that will be a new reality, and okay, the tenters of the patent will have, the right to explore, to sell the product. Okay, if that happens someday, Harold and Akshay will have the patent. That's fine. But as I said, we don't know if the patent will be granted to them, and people are dying now.
Mhm.
And we have to know if it can be replicated. And I wish Harold the best, and we have to respect his secrecy. But we are different people, and we will follow our own path, and be friends of him, no problem at all.
Yes, thank you for that response. And I'd like to highlight a comment from Daniel Tweed, who writes, longevity is its own reward. And I think many of us, especially those who aren't seeking to earn an income from longevity experiments, who are just seeking to have them happen, have that attitude, because one can do other things to make money. And that is my view. As long as one has health and youth and energy, one can do other things. And Corche also writes in the comments, any investment can be amortized over an optimal lifespan. So the longer you live, also, the more chances you have of earning a return on any investment that you make. And in the meantime, Super Thin Guy writes, aging is a terrible thing for everyone involved. And that, I think, is the priority, to overcome that terrible thing. Now, David Wood does have a follow-up question, and I think you partially answered it. His question was, would the patent mean that researchers such as the two of you who use E5 in your work, or who would use plasma derived from young pigs, may need to pay licensing fees? But per my understanding, the patent has not been granted yet, so there's a time window at present in which you wouldn't have to worry about that, and you could proceed with the experiment. That would reinforce the need to do those kinds of experiments sooner rather than later. Would you agree with that outlook?
Yeah, so there is this aspect, and as well as we are a non-profit entity, as far as we know, the patent, the licensing fees is only if you seek profit. So as we're doing this non-profit, the university is also not profiting from anything. As far as I know, there would be no fees related to that, because we're just trying to develop this. But if our side of the research led to a new intellectual property, or a new patent or something like that, I think then something like that, like a licensing fee, would be related to that. But we're just trying to reproduce it in a non-profit environment.
Yeah, even the agreement with the university specifies that there will be no intellectual property arising from this. And we asked it specifically, we don't want any intellectual property. I mean, I'm not sure what Nina said. I mean, if only when you make some commercial, if you sell some product, you need the authorization of the detentor of the patent. Maybe there are. I already read there are. This is a very blurry case, in which only if you make a product and sell, you have to have the authorization of the patent, or if you do it for your own use, you need to do to the authorization of. If you just do research, I mean, but as Lina said, we are a non-profit entity, and we are just reproducing a scientific article published in a scientific journal. Neuroscience is the official journal of the American Aging Association. It's a public article, received a lot of attention, including from neuroscience readers. They received it because of this article, Harold, like a prize of attention. Like, this article was very read in neuroscience. So it's a public article that, and in science, reproduction is essential. I mean, that's it's not because you publish an article that it's true. It's after this article is published and is reproduced and confirmed for other groups, even if the technology is a bit, it's not exactly the same, but only that it's transforms in true. So what we are doing is to checking if the article is reproducible. And so, in fact, in certain sense, we are doing something that, to be honest, it seems of interest of the company of Juven. I mean, we are taking an article and an experiment, and if it's reproducible, I suppose that it's very good for the company. They can say to the U.S. Office Patent, "Hey, our experiment is rejuvenating rats around the world. This lab from Brazil has immortal rats in their lab, and you won't give us the patent." So I believe that this can't help the company, because they don't have the patent. I repeat, they are trying. If you go to the public documents of the patent request, you see they are trying once again, and they try, and they are denied, and they try again. So our experiment could serve as an argument, like this is most successfully ever in biological sciences, maybe, and you won't give the patent to Harold and Akshay. So and there is another point. People are dying. I mean, people are dying now. Besides all what I said, besides all of that, people are dying. The investors of Juven are dying. The Harold Katcher is dying. Akshay is dying. My mother is dying. You are all dying. So if the patent doesn't exist yet, okay, let's advance it. And if the patent exists someday, okay, they can make the profits. I don't care. I will be happy to see them billionaires if everybody is rejuvenated. But we can't wait. We can't wait. People are dying now in the hospitals, in the nursing homes.
And
I concur.
One important thing that I don't know if we mentioned it is obviously it's good for Juven if the results are positive, if we are able to reproduce it. But if we are not able to reproduce it, I think it's bad news for them. But one thing that we want to do in this experiment is to be completely transparent with the results. So if the results are bad, if it's bad for the animals, or if there's no sign of rejuvenation, we want also to publicize this information, to publish an article. If it's a bit harder to publish like a negative result scientific article, but we also want to do something like that. We want to be completely transparent, like it didn't work, we weren't able to reproduce it, So this is also something that it's important to mention.
Absolutely. And publishing negative results is crucial for science as well. This was a fascinating conversation. Unfortunately, we are out of time for today's virtual Enlightenment Salon, but here I've posted again the link to make a donation to the Rejuvenation Science Institute. Again, large donations are welcome, but small donations are also welcome. Whatever you can afford. This experiment has a very modest budget as far as experiments go, so $75,000 to do the initial experiment, a bit more if you want the longevity study. I certainly want the longevity study, but please help out the Rejuvenation Science Institute. I agree, we are all aging and dying, and this needs to stop so that we can all live long and prosper.